I'm delighted to introduce our first plenary speaker for today, Professor Lynne Sneddon. I'm sure many of you are already really familiar with Lynne's incredibly impactful work in the field of fish health and welfare, particularly when it comes to pain and pain receptors, and are looking forward to hearing her speak today. Now, today, Lynne's going to be focusing on how empirical data can influence science, animal care, industries that use aquatic animals, government and regulatory bodies. She's also going to discuss engagement with media and how this can drive dissemination of information, and how scientific findings can be used to change public opinion. Now, I'm really looking forward to Lynne's talk, and I'm sure many of you are too. So now over to you, please, Lynne. You can share your screen and hopefully everything's ready to go.
Great. Thank you very much, Kate, for that wonderful introduction, and a big thank you to the organisers. I'm absolutely delighted to be part of this meeting. It's just so interesting and fascinating. And I think it is important that we show to the public we are value for money, and that the funding we receive through taxes actually makes a difference. And we use the science to improve the way that we do our science, and it has an impact on the animals' lives. And of course, I've focused on aquatic animals for my career, and I'm based at the Aquatic Animal Welfare Group. So hello from Gothenburg in Sweden, wherever you are in the world. And in effect, we have quite a complicated relationship with aquatic animals because we use them as a foodstuff. So we farm them in huge numbers. You can see the salmon here. And we also catch about 1 trillion fish from the wild every year. And now 50% of those fish are actually used to feed other animals. So for example, with our salmon, we need 1 kilo of wild fish to produce 1 kilo of salmon. So that doesn't seem to be a very sustainable practice. We also keep fishes as beloved pets, as a companion animal. And actually, fish are the 3rd most popular pet behind cats and dogs. And so we're taking these animals into our homes. And many people like to go and look at fish. There are proven psychological benefits of watching fishes and other aquatic animals. And there is an educational value for people who live within an inner city to actually go to a place where they can actually see fish, crustaceans like crabs, and actually learn about these animals' lives. And some people enjoy the sport of angling or use it as a pastime, a hobby. They like to go out as an individual and capture animals, very, very popular. But of course, that involves hooking them and taking them from the water where they suffocate. And so there is some ethical issues there. And finally, we use fishes in experiments. They are an important model for a variety of topics across many different contexts, and have become increasingly popular as a model. So in each of those contexts, there is the possibility of the animals to experience poor welfare. And so I personally think it's important that we understand how we treat these animals, how it affects them. I'm also very interested in other species of aquatic animals, the decapod crustaceans, which you can see here, crabs, lobsters, shrimps, and prawns. And of course, there's been a lot of media attention on decapod crustaceans lately. Many countries have banned live boiling of these crustaceans. And of course, we'd never dream of boiling a fish or a cow or a chicken alive, but yeah, we do it to these animals. And also the cephalopods, these are octopus, squid, cuttlefish, and nautilus. They are actually protected under European legislation for science, whereas the decapod crustaceans aren't. And that's thought to be because they are highly intelligent, and there is evidence for pain experience in these animals. And so I'm going to focus on fishes, decapods, and crustaceans. And the reason for that is because I work on them. But of course, marine animals, the mammals, seabirds, and all the other groups of aquatic animals are equally important. But today we're going to focus on 3 groups. And I'm going to talk about how basic empirical science can actually have a profound effect on the way that we treat these animals and can really drive the agenda with improving the welfare of aquatic animals. Now, there's a great focus on pain and suffering, and why might that be? Because, of course, if you show that an animal is capable of experiencing pain, and that is a very negative state for the animal to be in, then that really is powerful evidence for legislators, governments, regulatory bodies to change the way that we treat that animal. So there is a real focus on the sort of negative welfare aspects of science. Does this animal suffer? If so, then that's a very powerful statement to effect change in the way that we treat that animal and to protect them under legislation. But we shouldn't forget that positive welfare is just as important, and that actually we should be investigating what is it that makes welfare good in a fish or a crab or a cuttlefish. And we can refer back to Marion Stamp Dawkins' definition of welfare, which is really basically, is the animal healthy and does it have what it needs in its environment? And thinking about that, we can investigate, does the animal in the captive situation have items, enrichment we call it, in their habitat, in their cage, in their tank, that they actually need to perform normal behaviors or that reduce stress? And so that's the two kind of topics I'm going to focus on today, which is how does the study of pain affect the way that we treat animals, but also can we try and use strategies such as environmental enrichment to improve welfare? And I'm going to focus on two contexts. I would love to spend the next 10 hours talking to you about all of the contexts, but I'm going to focus simply on the use of aquatic animals as experimental models and how we can improve the welfare of fish and crustaceans and cuttlefish in aquaculture. Now, I started working on animal welfare in 1999, would you believe? And I went to the Royal Institute and worked in Mike Gentle's lab. And Mike was very famous for his work on bird pain. And he actually showed that the beaks of birds have pain receptors called nociceptors, and that when you cut the tips of the beak off with a hot blade, that was actually painful for the animal. He also showed that there were these nociceptors in the legs of chickens, and that when you shackle them, that's actually painful. But one of the great lessons that Mike taught me, he was a wonderful mentor, was that in order to drive changes in the welfare of an animal, you actually have to study poor welfare. You have to cause poor welfare in order to study it, because you need sound scientific data to go to a government and say, look, here, this animal experiences poor welfare, we should be protecting it. And there is a real focus on that suffering aspect, but I think things are changing for the better. But when I went to Mike's lab, we just really didn't know whether fish were capable of pain perception. No one had even investigated it. Now, just to start off with some definitions, you often hear the words nociception and pain used interchangeably. There is a precise definition of both. Nociception is just a simple detection of a stimulus that could or does cause tissue damage. And it is usually accompanied by a reflex withdrawal response away from that stimulus. So if you accidentally touch something hot, something sharp, you instantly feel that and you move your hand away. That's nociception. Now, if there's no damage to you, then that's fine. You don't experience pain, because pain is linked to the actual tissue damage. So it's both that sensory nociception experience and the psychological negative affective component of pain, the suffering and discomfort. So if you actually damaged yourself, you know, you start, that feels bad, it throbs, you change your behavior. And so it's both the sensory and psychological experience. Now, some people say nociception is distinct from pain. That's not the case, because you can't have pain, sensory pain linked to damage without nociception. But this is really key to driving changes in legislation and guidance. You have to really show that the animal should be protected because it is capable of experiencing poor welfare. Now, we talked earlier about the use of fish. We use them as a foodstuff in farming, aquaculture. We catch many, many fish from water bodies. Some people use them as a pastime, a hobby. Some have a beloved pet, and some people, like myself, use them in experimentation. And all of those contexts, there is the possibility of tissue damage to the animal. And so there is the experience of pain is going to happen in many of these situations due to tissue damage. And so in my view, that it's important that we understand what those situations are and can we do something to reduce pain or alleviate it in some way. Now, before I first published a study in 2002, we really didn't know whether fish had nociceptors, these receptors for painful stimuli. And the very early work I did looked at the neuroanatomy and the electrophysiology of nociceptors on the head of the rainbow trout, which you can see here. And this was the very first characterization. These are polymodal nociceptors. They respond to mechanical, chemical, and heat pain. And as you can see, they're on the face of the fish. And that was the very first publication that these animals had nociceptors. So there was a possibility of pain in fish. And that was quite a step change in the way that people thought about fish. I had many friends who were vegetarians at the time who would happily eat fish because they thought they had no feelings. And we also did further work where we characterized nociceptors in a variety of different ways. Here you can see chemical nociceptors that respond to acetic acid. That's like vinegar, if you've ever gotten vinegar in a cut, or carbon dioxide, which causes a peripheral tissue acidosis, so excites nociceptors. And this is citric acid and citric acid buffer. So you can see there's a huge number of these nociceptors on the face of the fish. And that then leads us into looking at whether there are behavioral changes. Are there signs indicative of a pain experience? And this is a very early experiment I did and published in 2003, where I looked at the time to resume feeding in animals which were controlled. These were rainbow trout, so nothing was done to them. Animals which were injected subcutaneously under the skin and the frontal lips with saline, which is a non-painful stimulus, it's just a physiological salt, or morphine administration, or they were given acetic acid injected into the frontal lips, or they were given the acid plus morphine. And morphine is a powerful painkiller, so it removes the pain. And what you can see is the time to resume feeding from the injection in the lips. And we can see that the controls, the saline injected fish, those treated with morphine, resume feeding within 80 minutes after the treatment. But the fish injected with acid took around 180 minutes. And it wasn't really until the sort of behavioral and physiological effects subsided that the animals resumed feeding. So there was a suspension in normal behavior. You give them morphine, take the pain away, and you don't see that suspension. We also looked at some unusual behaviors, such as rubbing the frontal lips into the tank. And with acid, you can see there's a high amount of rubbing behavior, but you give morphine and you see a significant reduction. And this work has also been reproduced in goldfish in Don Stevens' lab. And so we have these behavioral changes, which would suggest the animal is adversely affected by that noxious, potentially painful acetic acid injection. And I have to say that was quite revolutionary at the time because people really didn't think fish had any capacity for pain. And this is a study done by Lambert and colleagues where they looked at the number of papers which investigated the welfare of fish, specifically stress, fear, anxiety, and pain. And since 2002, the first publication of nociceptors, you can see there's kind of an exponential interest in the welfare of fishes. And I'm not suggesting causation, but I think that did change people's attitude to fish, that actually we should care about fishes. So that was the very early work that actually did then drive changes in the way that people thought about fish and the way that they were treated in a variety of different contexts. If we are using fishes, I believe morally and ethically, we have a duty of care to protect their welfare. And of course, in science, we apply the 3 R's, and I'm sure you're very familiar with those, but one of the R's is refinement, and that is the improvement of animal welfare. And of course, to be able to recognize pain, we have to develop behavioral indicators to do so. And then when we recognize pain in the animal, we should provide pain relief if we can't avoid that painful situation. So I'm going to talk about some work we've been doing on laboratory zebrafish. They are probably the most used model across Europe. About 50% of fish numbers in Europe are zebrafish. And I'm going to talk about some recent work we have ongoing on crustaceans and in cuttlefish. So the first study I've talked to you about, it was really trying to find a way of assessing pain in fish because they are such a popular experimental model. Fish are now the second most popular model across Europe. We use more fish than rats in experiments, and only mice numbers are higher. And this increase in the use of zebrafish probably reflects what's happening across the globe. The zebrafish, as you can see here, is increasingly being used in a variety of different contexts by biomedical scientists. And they're often held in these very barren tanks in these racking systems, so you can keep a high number in a very small space. Now, of course, for me, it's a no-brainer. We should be ensuring that our animals are in great welfare, at least at the start of the experiments, because that means then our data that we collect is valid and robust. But we don't really have many tools for fish to assess their welfare, and this would represent a really important refinement in the way that we treat fish in the laboratory. So I'm going to tell you about this project funded by the NC3Rs in the UK, where we sought to develop an automated artificial monitoring tool to accurately gauge the welfare of laboratory zebrafish. And then we used that tool to test a range of pain-relieving drugs, analgesics, via immersion by just dissolving them in the tank water. These fish are tiny, they're like about 1 gram. And so injecting them is quite invasive. So if we could just immerse them in the pain-killing drug, that would be an important refinement. And so what we did is we developed 3D tracking software in-house. What you can see here is the top view where you can see the fish is being tracked with this red dot, the side view where the fish is being tracked, and then we get 3D rendering of the behavior of the fish. And we can use this tool to measure up to 20 different behavioral parameters. I do apologize for the quality of these videos. We have to turn the resolution down to get real-time data acquisition. And we also developed a system for groups of zebrafish. This is much more complicated to get the data from these animals, but we could indeed get quite interesting information from that. What we did then is monitored zebrafish and undergoing laboratory procedures. We obviously had an undisturbed control. We had sham-treated animals which were anesthetized, but nothing was done to them. And then we had this range of procedures which are very common in the lab. So for example, fin clipping, the animal's anesthetized and then part of its tail fin is cut off to genomically screen the animal. We also looked at pit tagging, where the animal's again anesthetized, and this pit tag, as you can see here, is inserted into the abdomen of the fish. And then we looked at muscle damage after surgery, and then we compared it to a standard pain test, acetic acid, which we injected subcutaneously into the frontal lips. These are all done under anesthesia, but after the procedure, the animals are not typically given any pain relief. And so we looked at what were the behavioral indicators of healthy zebrafish. And these are individually held zebrafish. So what we see is in a healthy fish, it's continuously swimming. They swim in midwater. They use all of the tank space, calm swimming with quite gentle turns. When the fish have been subject to a laboratory procedure, we can see the fish is immobile for a prolonged period. They don't do very much. They use the bottom of the tank, or they don't move very much from a specific area. We see bursts of erratic swimming, and we see unusual behaviors that we don't see normally. And so here's some examples that, again, apologies for the resolution of the video, but the animal's being tracked, and you can see it's swimming in mid-water, a nice calm turn there. It's swimming constantly moving. And of course, that fish is a normal, healthy control fish. Now this fish, the video has started. The fish is sitting on the bottom of the tank. It's not swimming very much. It's wafting its tail a lot, but it's not actually going anywhere. This fish was fin clipped, and so you can see that the behavior is quite different. In this example, we have another fish which is in the bottom of the tank. It swims quite a bit, but then it sinks, which is a very unusual behavior for zebrafish, and then it almost bounces on the bottom of the tank. Very odd. That fish had a pit tag inserted into the abdomen, done under anesthesia, but no pain relief is given. And then our final example, here we have the fish in mid-water. It's swimming constantly, moving around, using all of the tank space. And this was a fin clip fish, but we gave it a local anesthetic, which has pain relieving properties, lidocaine at 5 milligrams per litre dissolved in the tank water. And you saw the behavior was much more normal. And what kind of data you can get from these? This is an example of a fish. This is a control. So you can see the diagram of the tank space. And this is 2 hours after treatment. And this is 25 minutes of movement. We have green dots, which are slow swimming, and red dots, which are fast swimming. And you can see the controls use all of the space. The sham handled, which were anesthetized, but nothing was done to them, used pretty much all of the space. But then we have the fin clip, and you can see space use is dramatically reduced, as is pit tag insertion, as is muscle damage, and this is 1% acetic acid. So the behavior is quite dramatically different. We then used the fin clip model, and we looked again at 2 hours after treatment, but we gave the animals pain relieving drugs. So we looked at lidocaine, a local anesthetic, which was very effective at returning behavior to normal at 5 milligrams per litre. Bupivacaine at 1 milligram per litre was not very successful. Flunixin, which is a non-steroidal anti-inflammatory drug, not very successful at 2 milligrams, but when we increased to 8 milligrams, the animals did use all of the tank space. And then an opioid morphine, 3 milligrams per litre, not very effective, but 48 milligrams per litre returned the behavior to normal in these fin clip fish. So we can use this information to then develop pain relieving protocols. We know which drugs we can use at what dose. We also looked at pairs, because of course zebrafish are held in breeding pairs, and what you can see here is a normal pair. They interact with one another, chase one another, and are constantly swimming. But if you fin clip one of them, and the one on the bottom of the tank has been fin clipped, you can see its behavior is very, very different. And it doesn't move very much. We also looked at groups, because of course zebrafish are held in as groups in stock, and this is the normal group behavior. They're all constantly swimming, they're all moving around and interacting with one another. And then if this one here has been injected with acetic acid into the frontal lips, 10% acetic acid, which is quite a high dose, and half of these fish actually sat at the top of the tank rather than the bottom of the tank. But as you can see, again, it's very, very mobile. So we gathered 20 different behaviors, and then we did a principal components analysis to see what are the most important behavioral signs that could be used for pain recognition in fish, in these laboratory zebrafish. And the first one was the distance swum. They swim much less. The location, they don't use the tank very much. They don't move a lot. And then activity was greatly reduced. And what we used was chromatic analysis. This is color analysis. And what you can see is this is red, green, and blue that is assigned to the data, and then it formulates a color basically telling you the welfare status of the fish. And we can do this in real time for individual zebrafish. And this is just an example of what we call the fish behavior index. So what you can see here is the graph tells us whether the animal's normal if the data is in green, okay if the data is in blue, unhealthy if the data is in yellow, and abnormal if the data is in pink. And this is the last 1 minute, last 10 minutes, 20 and 30 minutes. So it works backwards in time. This is a sham treated animal before anything is done to it. It's coming up as healthy, and 2 and 3 hours afterwards, it comes up as healthy. Then we have a pit-tagged animal. It was healthy. It went to okay because it was a bit hyperactive for a minute. So we really do need at least 10 minutes of assessment, I think, to get a reliable welfare status. But 2 hours and 3 hours after pit tagging, you can see the fish is coming up as abnormal. And so this is it in action. On the right here, we have the tracking. We have a normal fish. This is the health index. If you keep your eyes here, it's normal. We paused and then we fin clipped, and it will refresh in a moment. So keep your eyes on there. It's coming up as unhealthy. And when it refreshes after a minute of data, it's coming up as abnormal. So we can get a real-time welfare status from the software. And the software is free to download. We've published this, and my hope is that people are using it to improve zebrafish welfare. Now, one of the sort of more controversial things about this was that the fin clipping, lots of people tell me, oh, our fish are completely fine after fin clipping. We don't need to use pain-relieving drugs. But actually, there's been 3 other studies from different labs showing exactly the same results in relation to fin clipping, that the animals are negatively affected by that experience, that they do exhibit signs of pain. And also I've heard that we take much smaller fin clips than you did in your study, but that really depends on the kind of genomic screening technology that you have. And I know many labs that take around 25 to 30%. So at the moment, we're testing smaller fin clips. And so I have some examples here. Now, this is a fin clip of only 5%, and this is the behavior of the animal at 30 minutes. And what you can see is the animal stays in the bottom of the tank. So it's obviously affected by the fin clip, and it performs this kind of to and fro, quite hyperactive swimming back and forward constantly. So it actually becomes hyperactive. Then if we look at 60 minutes, the animal's doing very similar behavior, still staying in the bottom of the tank and swimming back and forth in this kind of hyperactive way. And then if we look at 240 minutes after the fin clip, you can see the fish is now swimming more calmly and is actually up in mid-water and is using all of the tank space. So what we found when we looked at the FBI assessment is that the 5% fin clip comes up as abnormal because of that hyperactivity and the increased use of the bottom of the tank for up to 2 hours. And that's similar to Nell tilapia, who increased their activity after a tail fin clip. But for the zebrafish, we do start to see recovery at 4 hours. So the smaller fin clip, the animals do recover much more quickly. And so it would be advisable to take this smallest amount of fin clip that you possibly can. Now, I personally would argue that 4 hours of pain is too much, and that we should use analgesia. We should provide pain relief to these animals, even if it is a small fin clip. Of course, if you accept the fin clip as painful, then you'd want to seek to replace it. And work by Kangwon Tilly and Will Norton's lab has effectively tested skin swabbing, and you can see that here. So what you do is you capture your zebrafish. These are sticklebacks on the right, and you take a cotton swab and you swab the animal along the length of the body in one direction. And they've shown that you get enough DNA from that, from adult AB fish, to actually perform genomic screening. We don't need to use anesthesia, which is a stressor in zebrafish. You must use a very light touch, 3 strokes. And so we collaborated with Kangwon and Will and looked at whether we could reproduce this in our lab here in Gothenburg. And that's exactly what we did. So we looked at using the FBI, and what we did was we looked at skin swab with no lidocaine, no pain relief, skin swab with lidocaine, fin clip with lidocaine. And much like we found in our previous studies, these all come up in the healthy range. But when we have a fin clip with no lidocaine, it comes up as unhealthy. And that's from 1 to 6 hours, really. When you take a 30% fin clip, the animal hasn't recovered at 6 hours. And then we looked at water cortisol. So fish excrete cortisol over the gills. So you can actually take a large water sample, filter it, and extract the cortisol. And what we see here is that cortisol was very high for fish that were clipped with no pain relief. But for the clip with lidocaine, swab without lidocaine and swab with lidocaine, you can see cortisol is much lower. And we've shown this kind of data before in other studies, so it's good to see that it was reproducible. And so now people are recommending the skim swabs, definitely for AB strain zebrafish that are older than 3 months. So that is quite an important replacement, and it's a good way of refining our experiments. Now, all of the data that I've shown you and data from other labs across the world have shown that a variety of different analgesic drugs actually do prevent pain-related responses in zebrafish. And you can see lidocaine, morphine, acetylsalicylic acid, diclofenac, flunixin. What you can see here in the doses are whether it's milligrams per litre, so it's immersion, or whether it's injection intraperitoneally, IP, or intramuscularly. And if you're interested in that, we have a recent review out that you can go and refer to. But what we have done is myself and co-authors have described the ways of recognizing pain in zebrafish, and we've suggested pain management protocols. And we say that any invasive procedures should use pain relief unless you're studying pain itself. And that is a working group report from the Federation of European Laboratory Animal Science Associations. So it's a European-wide report that should be uptaken by Europe, and we hope that other countries outside of Europe will follow suit. Now I want to talk about welfare of decapod crustaceans. We have a project funded by Formas in Sweden to look at the welfare of these animals. And as you know, they have 10 limbs. This is the Norway lobster, also called langoustine or scampi. What we wanted to do was actually electrophysiologically characterize nociceptors, because that actually hasn't been done in crustaceans. And then we wanted to look at the pain criteria. There are 17 criteria for pain in animals, and crustaceans so far only fulfill 14 of them. So we're going to look at the other 3. In effect, we want to develop behavioral indicators of pain and then determine which drugs prevent pain in these animals. And then use all that information to inform humane methods of killing. And this work is mainly done by my PhD student, Eleftherios. We had a master's student, Lisa, my collaborators, Albin, Peter, and Bjorn, and we're working with industry on this project. Now, we did studies on electrophysiology in shore crabs, and we recorded from the brain area of the crab, the G2 cerebrum, where tactile information enters the front area of the animal. And we applied different stimuli to the eye stalks, to the antennae, to the claws, and we used mechanical stimuli or touch. We looked at acetic acid as a noxious, painful stimulus. And then we used food odors and seawater as neutral stimuli, because nociceptors preferentially detect painful stimuli. They don't react to non-painful. And what you can see here in these lovely messy traces, this is acetic acid applied to the right claw, but this is the control. So this is water applied to the claw. So you see, if you see, these are the nerve traces. We're getting a sort of constant response. Then you apply 1% acetic acid and you get this rise in activity, which is characteristic of a nociceptor response. We then looked at mechanical stimulation. So again, we've got a control up here, nothing really happening. But then when you apply mechanical stimulation, you get this increase in response. So we found putative nociceptors because they respond to mechanical stimulation and the acetic acid, but they didn't respond to food chemical stimuli or to seawater. We then looked at the behavior of the Norway lobster because it's a very important fishery animal here in Sweden. And what we wanted to know is whether we could apply a painful stimulus, and we decided to use electric shock, if the animals react, and whether these responses are prevented by analgesic drugs. So we used lidocaine and aspirin in this case. So we had animals which were controls, nothing was done to them. Then we had animals which were shocked. Then we had animals which were injected with aspirin, but nothing was done to them. Then we had animals treated with lidocaine, but nothing was done to them. And then we had shocked with lidocaine and shocked with aspirin. And what we wanted to see is Does the electric shock, which is an acutely painful stimulus, affect them, and whether we could prevent that by using these drugs? Now, I hope this video doesn't offend anyone, but this is the animal responding to a 1-volt electric shock. So you can see there's quite a vigorous response. The animal responds in quite an elaborate way. Then if we look at this one, the animal's been given lidocaine. Now it does actually react to the shock, but we just don't see that really intense behavioral response. And that was the same with aspirin. We didn't see the intense response with aspirin. So those animals are responding, and we can prevent that by the use of analgesic drugs. We then looked at ways of killing animals, because of course all animals deserve a good death. They deserve a humane method of killing. And we looked at the signal crayfish, again, a very important animal which is caught for crayfish parties here in Sweden, and often they're cooked alive. So we tested methods that we thought would be more humane. We measured heart rate as a proxy for brain death because it's a neurogenic heart and the heart stops when the brain stops. We looked at live chilling in ice slurry. We looked at a laboratory method, which is overdose with clove oil. And then we looked at a very high electric shock. And we looked at, this is time to death. And so what we found was that clove oil took around 20 minutes for the animal to die. The live chilling in the ice slurry didn't work. The animals were still alive at the end. And then, of course, here we've got electric shock, which was really, really rapid. So we're using 100 volts here. And so the animal is dead very, very quickly. And one could infer, if your definition of a humane death is a rapid death, that electric shock is a better way of doing it. So all of those studies have shown us that the animals are negatively affected by a potentially painful experience, and it's important for us to think about the way that we treat them and keep them in good welfare and health, because of course we're going to eat these animals and we don't want to eat sick animals. We want to have sustainable populations if we're going to go out and catch them, and we need robust data on the animal if you use them as a scientific model. And obviously we need to safeguard our own public health and ensure the animals don't have parasites or any other pathogens. So there is evidence for pain. Bob Elwood has produced a huge amount of evidence in terms of behavior, and we've now tried to look more at the physiology of that. And it's important that we seek to improve the way we treat these animals in any context, whether it be the farming of shrimp or whether it be capture and fisheries. And we're working with our industry partners really to inform their equipment so that we ensure the animals get a humane and rapid death. And the next project I wanted to tell you about was the welfare of cuttlefish. Now, Robin Crook has done a huge amount of work on squid and octopus when it comes to pain. So we decided to look at cuttlefish as a different species, really. And this work was done by Tsu Hisin, our PhD student, and I worked with Joe and CC. And we looked at whether acetic acid resulted in pain-related behavior, and did lidocaine prevent that change in behavior. So acetic acid was injected into the arm of the animal, and some animals, they also got lidocaine to try and block the pain signals. And what we found was that the animals groomed the area a lot. I'm sorry, this is quite small, but you can see the animal throws the arm out quite a bit, and then it grooms it quite intensively where the acetic acid has been injected. And that grooming behavior has also been shown by Robin Crook in octopus. She's also shown other behaviors such as concealment and skin removal. So again, we have indicators there where we can assess pain in these animals in the lab, and then we can provide drugs that prevent the experience of pain. And that's really what the sort of work is all about, trying to find a way to assess pain so that we can improve welfare in both the laboratory, and of course this has relevance to veterinary care. We can then use this information, this empirical science, to then tell regulators, tell government bodies about the animal's experience, and we can of course engage the public in changing opinion. there is some barriers in terms of science where a lot of researchers are rather worried about the use of analgesic drugs and that it may confound their experiments. So we're now funded by the Swedish Research Council to take advantage of other people's experiments in a variety of fish species, and we will be giving pain-relieving drugs to some animals and looking at if there's any differences in the data. Now let's move on very quickly to positive welfare, environmental enrichment. And when I'm talking about enrichment, I'm really referring to items in the tank. I accept there's many other ways to enrich an animal's life. But typically zebrafish have been held in these sort of barren tanks with nothing else except themselves for company. But we've engaged in a variety of different tests, as has other people. And when you look at the real zebrafish environment, you can see it's very heterogeneous. This is a picture taken in their natural habitat. So in this experiment, what I did was I gave a fish which had been reared for 7 months in barren conditions access to 2 chambers, either a barren one or one which was enriched. And it's very small, I'm afraid, so I'll keep my cursor on it. But the doors are open for the fish, and then the fish can make a choice as to which area it's going to enter. And you can see it swimming and deciding, and it decides to enter the enriched area. And all of the fish exhibited a preference for the enrichment, even though they'd been reared up in barren conditions. And Paul Schroeder, who did his Doctor of Veterinary Medicine in my lab a few years ago, did some very simple preference tests where he took a normal zebrafish tank, put a divider in it with an opening, and then tested different resources against each other because he wanted to ask what the fish wanted. And he looked at the impact of sex, the impact of dominance relationships and pairs, and group preference. So we looked at pairs, a male-female pair, 4 males, 4 females in groups. And the animals were given substrate, either sand or gravel, or an image of both. And the reason for that is people don't want to clean gravel or sand in zebrafish facilities. And so we thought the image was quite a good solution to preventing extra time needed for cleaning. And we looked at submerged and rooted plants and also floating plants. And then we looked at an air stone as a sort of toy. And Paul created a preference order. But this is kind of what we did was we pitted things against each other. We did combinations. And thanks to Molly Millington for drawing this up for me. And this is a video showing you that 7 of the 8 fish are in the gravel side, with only 1 fish in the sort of barren side. And when we looked at the images of substrate, we found with a gravel picture, there was quite a definite choice over the barren area. So you can see the barren areas in this dark grey for males, light grey is the gravel picture, and both males and females choose the gravel picture area, whereas it was less clear for the sand picture. So this picture here I've sent to many researchers across the world, and what I still do is print it out, laminate it, and put it under our zebrafish tanks. I was very pleased to see that Klein Happel and colleagues at the University of Lincoln reproduced this work. So what they did is they had groups of zebrafish, and then they lifted a barrier and gave them access to an area with a gravel image. This is time from lifting the barrier, and what you can see is that the groups slowly move into the gravel picture area. In about 20 minutes, they are spending all of their time really within that gravel picture area. So it's nice to see that it was reproducible by another lab many years later. So I went to a commercial supplier and said, could you produce this? And they said, absolutely no way, we're not doing that. And lo and behold, we find that there are commercial solutions to this. You can buy these runners which fit under tanks, and there is a breeding tank which has an integrated gravel picture. Now, there's no money for me, I'm afraid, but at least on the website, the paper is cited, so that's good. But it's nice to see science informing this commercial development. What about aquaculture? Are they using environmental enrichment? What you can see is this study here. This is used in a fish farm with tanks, and what they're using is colored balls and fake kelp. And that's improving the welfare of juvenile salmon because they're not being aggressive to each other. So it's providing them with some enrichment. In this study, what you can see is the effect of environmental enrichment. So on the left here, there's no enrichment. These are the standard salmon tanks. On the right here, we have enrichment in the form of pebbles and trays and shelters. And the top images are 1 month-old fish, a few months old in the middle, and then in the bottom, it's 1 year old. So these animals were reared in these conditions. And then there was a disease outbreak, and the animals were treated for this disease at day 25. So here we have the disease outbreak, treatment at day 25. And what you can see is the solid lines are the fish that were enriched, and the dashed lines were fish that were not. And this is the survival. So survival at 1 is 100%. And then we have wild fish in black and hatchery fish in grey. And what you can see is that the solid lines, the enriched fish, have much better survivorship than the non-enriched fish. So again, evidence that the animals' welfare is improved such that they have a better immune system and can recover from disease better than those that don't have enrichment. Here's a very recent study on gilthead sea bream broodstock, where again, they provided very simple enrichment, these ropes in the tank. And what they found was that the animals, when there was structural enrichment, SE, the animals were much happier to swim on their own rather than swimming with the group. And that's a measure, really, when the animals are close together, it's a measure of anxiety. So it would suggest the animals were less anxious with the structural enrichment and were willing to independently swim both before feeding and after feeding, whereas the non-enriched fish NE did this much less often. So more evidence that enrichment in aquaculture improves the welfare of these animals. So I hope I've shown you that actually environmental enrichment, things in the habitat and the tank, are beneficial for welfare, both in laboratory fish and in aquaculture. So we get the science again informing practice. And actually we now have commercial solutions from, I've shown you a couple, there are many, many more, many other companies are producing in-tank commercial solutions that are easy to clean. But what we do need is more research to investigate specific species requirements and decide what is best for the fish to produce guidelines for a consistency and reproducibility between labs. Now, finally, I'm just going to talk very briefly about how do I disseminate this work. And I spent a huge amount of time on doing things which many people have told me are a waste of time, that I should spend all my time writing papers and getting grants. But for me, if the science doesn't have an impact, and the fact that I do study poor welfare, I think it's very important to communicate that in different ways and have an influence on the way that these animals are treated. So just very recently, about 2 weeks ago, I was called to Scottish Parliament online, of course, from Gothenburg, to talk about salmon welfare and Scottish aquaculture and what the welfare issues are. And I've also actually several times asked for a ban on the use of heat treatments and salmon lice, because actually the temperatures they're using are painful to fish. So I think it's very bad for their welfare. I also speak to the public as often as I possibly can. I did this last month, TEDx Stockholm, where I went and talked about why we should care about aquatic animals. And it's very rewarding to talk to the public, and many of them were not aware of the information on pain in cephalopods and crustaceans. And I speak to lots of journalists about this. So here I'm talking about fishes and the fact that we should be killing them humanely and giving them a good death. I talked to them about cephalopods. This is the octopus work that Robin Crook did on emotional pain. And I, of course, talked to local journalists. If any of you can speak Swedish, you can read this. But Swedes eat huge amounts of crustaceans, and often they kill them, boil them alive, or barbecue them alive. And so I talked to them about doing that better and using more humane methods. And so I think it's really important that we are thinking about aquatic animals and giving them the same respect as we do to mammals and birds on terrestrial context, whether it be laboratory, whether it be farming, whether it be any of these concepts. Good welfare results in a healthy, disease-free animal. The animal has good growth and it reproduces. And that is so important in so many different aspects of the way that we use fish. If you want to have a very diverse, healthy ecosystem, then we must ensure the welfare of wild fish. If you want to have sustainable populations and fisheries, again, we need to consider the welfare of those animals. And certainly in aquaculture, if you want the animals to grow well, be disease-free, you should ensure their welfare is met. None of us want to eat sick animals, and we know that we can catch diseases from them. So again, this is important for our own health. There are so many benefits to taking care of the welfare of aquatic animals, whether it be for society in terms of health, or whether it be an economic benefit to those industries that use fish. So my main message is that we should start applying the same legislation and guidelines that we apply to mammals and birds to aquatic animals, fishes, decapod crustaceans and cephalopods that I've talked about, but there are many other species that should get the same protections afforded to them. Just to quickly finish by thanking everybody involved in the zebrafish, cuttlefish and crustacean projects. Thank you very much for your attention, and I'd be happy to answer any questions.
Thank you so much, Lynne. That was fantastic. And there's lots of questions in the chat. But I'll just start with a brief one, if I may. So being at the forefront of such research, how did you find it when you started back in the early 2000s? Were there lots of challenges or difficulties you encountered? Was it difficult to get support?
So I think, in terms of teaching, you'd ask a group of students who thinks fish experience pain, maybe one or two would put their hand up. Now you ask them and pretty much all of them put their hand up. So it's been quite a development in people's attitudes to fish, but it was quite difficult initially. I mean, most of the industries that use fish were not very happy about this. And so I remember going to speak to the UK Parliament in London and having a lot of very angry faces looking at me when I was talking about this. So I guess I've had to be brave over the years, and really I focus on the science. I say, here's the evidence, and then you need to decide really ethically and morally what you do with that evidence. And of course, I can provide an opinion on that, but it has been challenging in terms of other scientists as well. When I submit grants or papers over the years, I was never allowed to call it pain for quite a long time, because some people were just totally opposed to the idea of fishes experience pain, and they would write the most aggressive, unpleasant reviews. So it was quite difficult and challenging. It's much, much easier now, and I can use the word pain when I write papers and grants, and I think it's very much accepted now. But it can be quite a struggle to get people to understand that the science, if I was showing you this in a mammal, everybody would be perfectly happy with it. But for some reason, there is an element of speciesism towards aquatic animals, and that's a bit sad. So it has changed dramatically, and I'm pleased to say it's much easier now.
Thank you. A question from Elodie. Is there any clear evidence in some species suggesting that nociception can exist without pain?
Yeah, so you can have nociception. So if an animal encounters anything that could potentially damage it, and it senses that and it withdraws from it quickly enough, and there's no damage to the animal, then the animal doesn't experience pain. It's a very difficult question, because of course I've never been a fish or a crab in a previous life, so I don't truly know what they experience. What I can tell you is that the animals are adversely affected, that in the fish studies that we've done, you can't divert attention away from the pain. When they're in pain, they're really dominated by it. And so the mechanisms seem to be evolutionary conserved. There's certainly a lot of work in Drosophila, C. elegans, Aplysia, the sea snail, that show that they have the basic components for nociception and a pain response. But how does the animal experience that? It's very difficult to say. So you can have nociception without pain if there's no tissue damage.
And another question from Sarah related to your tracking software. She just wondered whether it's been used for recognizing stereotypies.
Yeah, I mean, it can do, and some people have commented on the data that it does look like the fish are kind of swimming back and forth, so quite in a stereotypical sort of way that we classically see in zoo animals and other species. So we sort of can measure up to 20 different things. We haven't really interrogated the data with regards repetitiveness of the behavior, so we haven't actually looked at the data. But if you know, if you're interested, I would be happy to send you the data if you wanted to look at it. But yeah, it would be another thing that we'd have to add onto the software, really.
A question from Elena. Why would anyone deny experimental fish pain relief?
Yeah, so it's very much down to an opinion that you must have a human-like cortex, a forebrain like humans, to experience pain. And if you accept that definition, then only really primates and humans can have pain. That's really the crux of the argument. So skeptics will say you must have specific parts of our highly developed 6-layer cortex. Fishes and birds have a 1-layer cortex, a 1-layer forebrain. And so they say that because they have such a simple area, that it's not possible for fish to consciously experience pain, know that they're in suffering. Unfortunately, there's been a recent study that's shown that this is not true, because when there's a patient who has damage to the areas that the skeptics say are important, it's the anterior cingulate cortex and the insula of the forebrain that they believe is the important parts for experiencing pain. And this patient has had a disease that's destroyed those parts, they're not functional anymore, and yet they still experience pain. And actually they're actually hyperagesic, so they actually feel more pain than they did before. And so many scientists think pain is actually subcortical, it's below the sort of forebrain, but the forebrain thinks about it and puts it into context. So that's what it comes down to. It comes down to brain anatomy. The problem is, you know, that by that reckoning, fish shouldn't be able to see because they don't have our visual cortex. And of course, fish can see. So it just really is a sort of, I guess, a semantic problem, and the skeptics define pain as having a human-like cortex.
Another question from Crustacean Compassion. Do you have any thoughts on the lack of inclusion of decapods, the only sentient beings to not be included, in the UK's Animal Scientific Procedures Act, especially with sentience being recognised legally in 2022, and your professional opinion on this?
I do think there is enough evidence to suggest that there is a pain experience in decapod crustaceans, so they should be included in the scientific legislation, and that conversation's going on in the UK and Europe wide. And I think that there is an issue, and it's more of a practical issue, that we don't have a lot of evidence on the welfare of these animals, and so how to keep them, how to kill them. And so I think they'll probably, it'll take some time for them to get put in because of those logistical difficulties. But really, there is no reason why they shouldn't be put in at this point in time.
And then just following on from that, there's a question / comment from Helen that I'll just kind of summarize about what do you think has delayed progress in that way? So it's always such popular media subject when new research comes out, but it's taken serious amounts of campaigning to even make small changes or wins.
Yes, and I think it's because the industries that use these animals are very powerful, and in effect don't want change. A lot of the industry sees welfare as something that's going to cost them money, when in fact I always try to convince them the opposite is true. If you have animals in good welfare, they're disease-free, they're growing really well, they've got great reproductive output, it would benefit you to you know, improve the welfare of these animals. But there is a lot of resistance because people think it might cost them their profits, that it might eat into the profits. And these groups that use fishes and other aquatic animals are powerful lobbying groups. And you know, for example, in the UK, there's 5 million people who like going out and catching fish. You know, these are powerful lobbying groups, and so the governments are rather reluctant to make major changes. And I know when the fish pain work came out initially, 2002, 2003, the government basically said they would wait and see what Europe did. And they decided they wouldn't change anything, because obviously fish were in the scientific legislation at that time, but they weren't in any of the other legislation. And of course, obviously, the UK has left the European Union. So it is really about this conflict between industries that use the animals and the science. And so there is a big delay. It takes a lot to make these changes.
Thank you so much. I'm sure we could keep chatting for a very long time, but in the interest of time, I'll hand over to Hannah to introduce our next speaker. So thanks again for such a fantastic plenary and a great way to start the day.